An enlarged heart. That phrase can mean two wildly different things. On the one hand, it's the super-tuned engine of an elite athlete, a sign of peak physical fitness. But on the other hand, for millions of people, it's a terrifying warning sign of heart failure. They can look almost identical from the outside, but inside, they're running completely different software. So today, let's crack open this genetic mystery.
So here's the breakdown. On the left, you've got the athlete's heart. It's a good thing. It's an adaptation to exercise that just makes the heart better, stronger. But then on the right, you have the diseased heart. This is a bad response, a reaction to stress that can lead to, well, disaster. So yeah, they're both bigger, but one is a finely tuned race car engine and the other is an engine that's literally about to break down. And that really is the million-dollar question, right? If they can look so similar on the surface, what's the real difference deep down? A simple physical exam might not tell the whole story. We've got to go deeper. I mean, way deeper. Past the muscle, past the tissue, right down to the genetic code that's running the whole show.
And to do that, to solve this puzzle, researchers needed a special kind of tool, a new kind of genetic lens, you could say, one that could let them peer right inside the heart cells and read the secret instructions they're following. That tool is called RNA sequencing, or RNA-Seq for short. Now, think of it like this. Your DNA is this massive master cookbook stored away in a library. It has every recipe imaginable. But the RNA, that's like the individual recipe cards that have been pulled out and are being used in the kitchen right this second. So RNA-Seq basically lets us peek into the kitchen and see exactly which recipes are being cooked up, giving us a perfect snapshot of what the cell is actually doing. And you have to understand, this was a huge leap forward. The older methods, they were kind of like trying to figure out what's in a giant library, but you were only allowed to look for books on a specific pre-made list. But RNA-Seq? Whoa. It's like getting the key to the entire library. You can see everything. It's way more sensitive, way more accurate, and you can even find brand new recipes, new genetic activity that nobody even knew was there before. Okay, so once scientists pointed this powerful new lens at the problem, the very first thing they found was, well, it was kind of mind-blowing. It wasn't just which genes were being turned on or off, it was the sheer volume of the genetic chatter.
So get this, in a healthy, enlarged heart, the athlete's heart, the response was incredibly precise, super controlled. Only about 245 genes changed what they were doing. It's like a focused, efficient tweak. You could almost call it a genetic whisper. But in the unhealthy, diseased heart, it was total genetic chaos. A whopping 2,041 genes were changing their activity. That is more than 8 times as many. So this wasn't a whisper. This was a full-on, desperate, panicked scream at the genetic level. And it gets even crazier, because it's not just about the number of changes, it's about the direction they're going. Look at this. The diseased heart is just frantically turning genes on. 1,429 of them. It's like it's flipping every switch it can find. But the healthy heart? It's doing the exact opposite. Its main move is to calmly turn genes off. It's a much more controlled, suppressive response. And if you think that's strange, listen to this. The diseased heart isn't just turning up the volume on genes that are already playing. It's actually waking up 417 genes that are supposed to be totally silent in a grown-up heart. It's almost like the cells are in such a panic that they're digging up old, forgotten instructions from when the heart was first being built. Okay, so the first clue was about volume, shouting versus whispering. But the second big clue they found was way more subtle. It wasn't just about turning genes on or off. It was about how the cells were literally rewriting the recipes as they went. This process is called alternative splicing. And that recipe analogy we used earlier, it's perfect for this. So if a gene is the recipe, alternative splicing is like a clever chef who takes that same recipe and says, you know what? For this version, I'm going to leave out the onions. Or for this one, I'm going to add extra garlic. You're using the same basic instructions, but creating slightly different dishes, or in this case, different proteins. So here's the interesting part. When they looked, they found that both types of hearts are doing this a lot. The diseased heart made about 513 of these little recipe changes, and the healthy heart made 414. Now, those numbers are pretty close, right? And that tells you something really important. The secret isn't that they're splicing, it's what they're splicing and how they're doing it. And this is a perfect example. There is a gene called FHL1. As you can see here, in the diseased heart, the cells are specifically using alternative splicing to cook up a different version of the FHL1 protein.
It's a very deliberate change, but in this case, it's a change that's part of the disease process.
So why does this matter so much? Well, because these seemingly small recipe tweaks can have huge consequences. You can create a protein that does something completely new. You can make it more or less active. You can even change its address and send it to a different part of the cell. It's an incredibly powerful way to fine-tune how everything works.
All right, let's recap. We've got our two big clues. First, the huge difference in the number of gene changes, the shouting versus the whispering. And second, the subtle but powerful rewriting of the recipes. So now it's time for the final verdict. Let's put it all together and see the unique genetic signature for each of these hearts. And boom, here it is. This crazy-looking chart is a heat map. But think of it as the final genetic fingerprint. It shows us everything at once. Each row is a different job the cell has to do, and the colors show whether that job is being ramped up or shut down. And you don't have to be a geneticist to see, just at a glance, that these two hearts are telling two completely different stories. So let's look at the story of the diseased heart first. What's its signature? Well, for starters, the basic stuff, like making energy and contracting the muscle, those pathways are getting shut down. It's like the engine is sputtering, running out of gas. Meanwhile, the immune system and cell damage alarms are going into total overdrive. And to top it all off, it's actively turning on the very genetic pathways that are known to cause heart disease. I mean, it's a crystal clear signature of a system in crisis. Okay, now let's look at the healthy heart's signature. It is a completely different world. It's smart. It's actively suppressing inflammation to prevent any unnecessary damage. And remember how it didn't just shout by changing thousands of genes? Instead, it intelligently fine-tunes things using those clever recipe changes, that alternative splicing. The whole response is just precise, efficient, and ultimately it's protective. And the scientists who did this research, they put it perfectly. They said that exercise gives the heart a more efficient way to send signals by using alternative splicing, rather than just changing the expression of a bunch of genes. In other words, the healthy heart works smarter, not just harder, or in this case, louder. So when you boil it all down, there's our answer. The critical difference between that athlete's heart and the diseased heart isn't the size, it's the software. It's the unique genetic program running inside. One is a program of smart, efficient, protective growth, and the other is a program of chaotic, panicked, and destructive stress. Which, of course, leads to one last incredible question. We can now read these genetic signatures in stunning detail. So, what if we could learn to rewrite them? Can you imagine a future where we could develop treatments that gently nudge a diseased heart's chaotic program and guide it back towards the smart, efficient program of a healthy one? Now that is the incredible future that this kind of research opens.